condensed chromosomes. This is just long spaghetti noodle DNA, extended form. It's just hanging out. It's not condensed, not taking the shape of actual chromosomes. Takes the shape of chromosomes, that's prophase, okay? Distinct chromosomes, replicated chromosomes. Lining up on the metaphase plate, centrioles are here, mitotic spindle is here. You see how they're all perfectly lined up in the middle. Middle is easy. Meta means middle. Metaphase plate, they're lining up on the equator of the cell, okay? Then, another metaphase here, they're starting to kind of get a little bit of tugging by the motor proteins and the enzymes. Here, anaphase are being totally pulled apart. Telophase and cytokinesis, you're getting the cleavage furrow. They're actually pinching into two distinct cells. They're about to completely bud off and have two separate cells. Yes? Good? Any questions about mitosis? Yeah.
What, do you need to know do we just need to know interphase, prophase, and metaphase, or do we need to know what each one does specifically?
You need to be able to know what each phase is. So looking at those different phases, like I just showed you, which phase is which, right? Non-condensed chromosomes just hanging out, doing their thing, interphases. Compact, discrete chromosomes that are they're disordered, but they're there and they're replicated, and then going through the stages of mitosis. Yeah, you should be able to see these. And in lab, I'm going to have different stages of mitosis set up where you can look under the microscope and look at real embryos and cells that are undergoing mitosis. So, stem cells are a really good example, a beautiful example of cells that undergo massive mitosis. They're undergoing mitosis all the time. We have in tissue development, the tissues are made of highly specialized cells that arise from 3 embryo germ layers. So early in the embryo, as early as 3 weeks of human development, way before this is about 9 weeks, 10 weeks. Early in development, at 3 weeks of development, the early embryo has sort of 3 layers. It's like a 3-layered embryo sandwich. I know that sounds weird. But that's how the tissues start to line up. They have a layer on top, a layer in the middle, and a layer on the bottom. There's nothing discrete that you can see except layers. Those layers are endoderm, mesoderm, and ectoderm. And if you look at these under the microscope, you'll know exactly what I'm talking about. They're just little layers. That is going to set up the future body plan and future tissues of that organism, of that embryo. And so early on, there's all these stem cells. The first one cell of the embryo is a stem cell. It has the potential to give rise to all these other structures and cells. So during development, you need stem cells. They're actually considered some of the true sort of mother stem cells are there right at the very beginning. They have an amazing ability to divide repeatedly, both in the embryo and in the adult. You all have stem cells in your bodies, and I'll show you examples of where they're heavily used. So what are they? What are stem cells? They're pluripotent. That means they have the potency or the ability to give rise to a lot of other cells. Okay? So they're pluripotent. They can give rise to lots of other cell types. If they get the right cues, they will. They're self-renewing. What does that mean? They're self-renewing. They actually can give rise to a daughter cell that's going to become differentiated and become a specific type of cell. But in the process, they will regenerate. That second daughter cell is going to give rise to another stem cell. You won't find this in your textbook, by the way. This is like my stuff. This is outside, this is research and my stuff in an area that a lot of books don't have a lot of information on. I mean, some, we can get a whole entire stem cell book on this now, but this is all my added stuff. So they're self-renewing. In other words, if I have a stem cell sitting in the bone marrow, for example, and I want to keep proliferating and making more blood cells, if I'm a stem cell, I need to self-renew. Otherwise, if I give rise to 2 daughter cells by mitosis, I just lost my stem cell. No more stem cells. So they have the ability to make one more of themselves and then make the daughter cell that's going to go on and give rise to the blood cell or whatever, muscle cell or tissue, whatever it's going to be. They're also immortal. That means they can divide an infinite number of times. They keep dividing by mitosis. Making daughter cells, they will divide. Making daughter cells, they will divide. Like that stem cell will keep on undergoing mitosis. This is how we can use stem cells to study all kinds of development and disease in the dish, and we can freeze them and thaw them and send them to other labs and keep using them for years. There's no need to keep trying to accumulate them from human beings because we can continue because they're immortalized cell lines. So why do we need them? Why are they important? Obviously for development of us, a multicellular organism, starts with one single cell, the egg, that gets triggered to develop and generate all the tissues of the body. If you think about it, you go from 1 cell to 40 trillion. From 1 cell. I mean, I don't know about you, but to me this just blows my mind. All this just, it's absolutely amazing. There's definitely some divine beauty that's happening at play here. Stem cells are central to that role in the developing organism. We also need stem cells for regeneration. Throughout your life, every single day, if you didn't have stem cells, you would actually die. So it's not just important going from 1 cell embryo to whole organism, but throughout your life to regenerate your cells and tissues. We lose millions every second. Okay, so here's some examples. Without a supply of stem cells, you would lose your intestine in 2 days,
Wow.
your skin in 3 weeks, and your red blood cells in 4 months. Bye-bye. No more red blood cells. No more gas. No more respiration and cardiovascular activity. You're dead. So stem cells are in your bodies in these pools and pockets of your tissues that allow all these organs to be regenerated every minute, every day. So how do we get it? And I know this gets into, for some people it gets into ethical and controversial and sometimes religious discussions and social aspects of the ethics. I teach a whole course on stem cell biology, so we're gonna spend just a few minutes in here. But I just wanna give you a little glimpse into where we get them from, because a lot of times people are kind of either for them or against them, but they really don't know the biology. So they speak without knowing any science or any biology at all, even the politicians, especially the politicians. It's like they have no idea. Where we get some of the human embryonic stem cell lines that are in existence today, and we grow in the dish and we use them for various experiments, we got them originally from donated embryos that were donated from couples going through IVF, in vitro fertilization. They have excess embryos. They had successful pregnancy, they did their family building, they're done, they have their embryos, and now they can either pay the clinics to freeze their unused embryos, and there's a freezing fee for that, of course, like all these things. Insurance doesn't pay for that, they have to pay for that. Or they donate them to a surrogate couple who's trying to conceive and can't, and would like donated embryos and eggs and gametes. Or they can discard them, and that means incinerate them. So the clinics will incinerate billions of these, just get rid of them, they burn them, okay? A lot of people are not aware of this either. They go, it's horrible, stem cells are terrible. Well, the scientists went, wait a minute, if you're gonna throw away or burn all those human embryos, if we could just get this 4 to 5 day embryo, we could take out the inner cell mass. We could actually suction it out, little pipette, just suction it out. This is the trophoblast, this is the inner cell mass. Keep in mind, this is about 4 to 5 days of human development, no bigger than the head of a pin. Take those cells out, we can actually plate them in the dish in vitro, and they will grow and they will continue to grow and divide. And they like to stay associated. They make like these colonies where they stay sort of connected, and they're self-renewing and they're pluripotent. They can be exposed to various different chemicals or hormones, and they can give rise to all the different tissues of the body. Even more recently, in the last decade or so, eggs, things like oocytes, can be generated from stem cells in the dish. So huge potential for development, regenerative medicine, healing individuals that have a lot of horrible ailments and diseases. This comes from discarded IVF clinic embryos. The couples were consented, so they chose. And I will tell you, the majority of couples will not choose to freeze indefinitely because it's expensive, and they will not choose to donate for surrogacy for whatever reason. I guess they want to think they have children out there in the world that are theirs. I don't know, a lot of people will not. So they'll do either incineration, discarding, or donation for research. And so that's where that kind of that aspect comes in. Okay, does that make sense? Okay, so they undergo mitosis and give rise to all these beautiful tissues. Current stem cell research, though, we can get stem cells, culture them in vitro, turn them into different cells, like neurons, for example, with growth factors, grow and characterize those cells. And the idea is now in clinical research to transplant those into patients that have different diseases. Parkinson's disease, spinal cord defects, cardiovascular disease to repair damaged heart tissue. I don't know if you guys know Michael J. Fox, you know him as the Parkinson's research spokesperson. He's Back to the Future actor in the 80s, so he's a big 80s actor. I really like him a lot. He's done a lot of advocacy and research and donations from himself going into the Parkinson's Research Foundation. But the idea now to even cool stem cell research is not just coming from human embryo stem cell lines that we have now frozen down, and there's lots of them. Don't have to get them right from embryo because the cells will proliferate immortally, right? The newest thing, though, is you get a skin sample from a donor. We can transform those adult cells with certain factors into embryo-like cells or embryonic-like stem cells. Those are called induced pluripotent stem cells, or iPSCs. And the idea for this is you would no longer rely, originally a long time ago, on the embryo that first provided the cells to have the cell line, right? No embryo was discarded or used in that process. And you would be making stem cells from the individual that you would likely, they would be wanting treatment with those ultimately. So in other words, converting things, and they do it by a biopsy. So you get a little biopsy of skin that has little fibroblasts and different cells in it. Put on a series of factors that actually convert those adult cells into embryo-like cells. And they look and act pretty much just like embryonic stem cells. And then those, you can culture into the different cell types of interest, like neurons, and do transplantation stuff. These are already underway. This is already being done. Now, I don't know if you guys realize, this sounds like sci-fi, if you really think about it. Because you're taking skin cells, you're turning it into an embryo-like cell, then you're transforming that into the cell type of interest. Now, what you prevent is immune rejection. That individual recognizing those cells as non-self and making antibodies against it. Remember the blood agglutination we did in lab? If you do not have that antigen, you have the antibody against that antigen. So if you get somebody else's stuff, protein, cells, you're going to have an immune rejection against it. Your body's going to try to attack it. This new IPS discovery in the last, since 2007, so it's going on about 12 years, now it's just exploded. There's all kinds of clinical studies that are looking at this right now. And just to show you how cool it is, we can take cells from an individual, skin cells. This was my friend So Young at Stanford, and we took these videos together. She donated because she thought, if I'm going to have subjects do it and I'm going to study it and make these cell lines, these IPS cell lines, I want to go through the process and see what it's like to do it. So she gave her a little biopsy, core biopsy punch. They do a little one stitch, so she has a tiny little scar. And then from those, she converted them into iPS cells, embryo-like stem cells. And then from those, converted those into heart-like cells. So I was joking around with her. I said, these are like So Young's mini hearts. They look like, I was like, oh, we're looking at So Young's mini hearts beating. I don't know if they're going to beat. These are in the dish, 37-degree incubator. Her skin cells, we turned into heart cells, and then the heart cells naturally contract and have a rhythmicity to them of about 60 beats per minute, which is about what the human heart is, 6 beats per minute. Crazy? Okay, those came from her skin. I'm like, so yeah, we're looking at So Young's mini hearts. I was so excited. So we were videotaping, and like, this is just one little sliver of where this research is going, and using these mitotically dividing amazing cells. We can study human heart in the dish, we can study human development, human growth, function. There's a lot of promise for regenerative medicine. Healing diseased hearts, spinal cords, brains, muscular ailments, all these different things. So I will skip, I'll let you do these as the homework part. So you asked Lewis, do we have to know the phases? Yes, you have to know the phase of mitosis. This question I want you to put in order from chronologically where they go from what stage to what stage as you go through mitosis. The second part is apply what you learned about cell structure, cycle, and mitosis. Come up with a hypothetical drug that would slow or stop the cell cycle. This has big implications. People have been working on this for decades. So it's not easy, but there are different ways. Describe exactly how it works, its target, what it might be used for clinically. So I want you to do those. You don't have to do the water, blood, IV one. Just do these last two to help bring all the mitosis, stem cell mitosis stuff into play. And how are we, that's perfect time. You've taken, do I have your cards? You have your cards. Yeah. No.
Yeah, I have some. You have ours.
I can't even remember.
I definitely have mine.
I have mine.
Okay, I'll see if I have them. I might have some yours. Oh, I do. I have several. I have a few. But you guys have the rest. I have Melissa and Alex. But I don't have anybody else's because we have the text. Did you give me one too?
No,
Oh, you need a new
I
one?
run.
All right, well, let's just, if you can't find it, I have new ones I can give out. Just jot some stuff down at the point, and I'll remember the other week this week. Okay.
Thank you.
Yes, yes. All right, have a good rest of your day. Oh, you have stuff.
Thanks, you too.